And in order for them to make sure that the pre-fusion conformation is stable and not change into post-fusion conformation, they added 2 proline amino acids. Yeah, so this is the structure of proline, as you can see here. So, sorry. Oops, sorry. Sorry, yeah. Okay. So can you see the slide?
Yes, Jatin.
Yeah. So basically, this is the structure of a proline. Yeah. So the reason why they add 2 proline is to increase the rigidity. Yeah, to increase the rigidity of the a spike protein, because you know that the proline, yeah, this NC bond cannot rotate. Yeah, this NC bond cannot rotate. So once you have proline in your protein sequence, the structure will bend. Yeah, the structure will bend like this. Yeah, if you have proline here. Okay, because of the uniqueness of this bond. Yeah, so you can see that this bond is not rotatable. Yeah, and most of the time proline will be in the trans conformation. Only in a very specific situation, trans conformation will change into cis, and it is unlikely to happen in normal situation. Yeah, so they will maintain as a trans conformation, and the protein sequence will bend. The structure of the protein will bend. So as you saw yesterday, yeah, the pre-fusion conformation is close. Yeah, dia macam apa, dia melipat, is folded. So this structure can be maintained if you have proline. So it will not open to become a post-fusion conformation. So that's the a very smart strategy. It's very simple and smart. It's very simple and smart, and they are able to do that because they have deep understanding about the properties of amino acid. So if you do research in protein chemistry, you will know. You will know that proline has this characteristic that could be an advantage for some in some cases. But in some cases it becomes disadvantage. For example, if we want to design a protein with alpha helix structure, because you learned yesterday that if you have a proline in your alpha helix structure, then the alpha helix structure will break. So dia ada beberapa keadaan yang dia you can use proline for your own benefit, for your own advantage, but in some cases you have to avoid proline because you know the properties of proline, it will break the alpha helix structure. All right, so this is gamma turn. This is gamma turn. This is another type of turn. It's different from beta turn. It's smaller. This is smaller. So it involves 3 amino acid. So kalau beta turn yang semalam, it involves 4 amino acid. Dia besar. Kalau gamma turn, it involves only 3 amino acid. So it's smaller. And the turn is stabilized by the hydrogen bond between amino acid number 1 and amino acid number 3. Okay. As you can see the structure here, this is the hydrogen bond that stabilizes the gamma. All right. So this is a method. We will discuss this in the next lecture. This is the spectroscopic method to see the secondary structure of protein, to see whether your protein has alpha, beta, or random. We call it as circular dichroism spectrophotometer. So we will learn this when we discuss about spectroscopic method for protein analysis. So basically, this is a typical information that you get from CD, from circular dichroism. You have alpha, you have beta, or you have random coil can be easily seen from the spectrum. If your protein has alpha structure, you will have this, this kind of peak, yeah, beta conformation, this kind of peak, and for random coil, this kind of peak. But in most proteins, they are a mixture between alpha, beta, and random. So the peak will become a bit complicated, but we have a computer program to calculate the proportion of alpha, beta, and random coil. So you can get the ratio, how many percent alpha, how many percent beta, and how many percent random coil in your protein by using the computer program, yeah, the algorithm in the computer that is connected to the spectrophotometer. Yeah, so so we can, the analysis is very quick, just like any other spectrophotometer. You just put your sample inside the spectrophotometer and you get the result within minutes. Yeah, but the information is very general. Yeah, you just get information about how many percent alpha, how many percent beta, how many percent random, but you don't get the atomic structure of the protein. Yeah, if you want to get the atomic structure of the protein, then you have to do X-ray or NMR. But of course, it takes time, maybe several months. Yeah, you need to crystallize the protein, lepas tu kena analyze pula. Yeah, tapi kalau CD, it's very quick. Yeah, so we use CD just like we use IR. IR kan cepat kan? IR is very quick, but the information we get also limited. Yeah, from IR, the information that we get is just what functional group that we have in our compound, whether it has ketone, it has aldehyde, it has aromatic group. So sama macam CD lah. The information that we get from CD is very limited. It's just how many percent alpha, how many percent beta, and how many percent random coil. But it could be a very useful information if you want to monitor the stability of the protein. For example, kalau you nak tengok sama ada vaksin lepas hantar daripada China datang Malaysia, you nak tengok sama ada vaksin ni still stable. Yeah, you can run CD, very quick analysis to see whether your vaccine is still in certain conformation. Yeah, for example, in beta conformation, masa sampai sini dia ada jadi random coil. Sebab panas kan, sepatutnya dia simpan dalam fridge, ini letak dekat luar. So the vaccine dia become damaged lah. So you can use CD, a quick analysis to see whether the protein inside the vaccine bottle already changed into random coil due to the high temperature. Yeah. All right. So and then a protein tertiary structure. Kalau you ada beberapa protein secondary structure, then you combine them into one whole protein. We call it as protein tertiary structure. Yeah. So actually, it's an overall spatial arrangement of atoms in a protein. And protein tertiary structure, as we discussed yesterday, I explained yesterday, they are stabilized by a numerous weak interaction between amino acid side chain. Yeah. As you still remember, hopefully, from yesterday's lecture, the weak interaction that is involved in stabilizing the protein are hydrophobic, polar interaction, hydrophobic bond, but there is a covalent bond. There is covalent bond called disulfide bond that also can stabilize the protein structure. Disulfide bond is a bond between cysteine and cysteine. So kalau you ada cysteine, ya, dia punya side chain dia ada SH, ya, dia ada SH, and you have another cysteine, ini pun SH. So if you oxidize them, yeah, so H ini hilang, so you will have a covalent bond between S. Yeah, so it will stabilize the protein tertiary structure as well. There are two major classes of protein tertiary structure, fibrous and globular. Yeah, fibrous protein ini selalunya water insoluble, dia hydrophobic. Yeah, mainly consists of beta structure. Ya, dia water insoluble. Contohnya rambut, kuku, lepas tu horns, ya, tusk, ya, they are proteins, ya. So it consists of your skin keratin, ya. Keratin is water insoluble, ya. That's why kalau you mandi kulit you ada lagi, ya. Kalau you mandi, kalau protein dia soluble, lepas mandi kulit tak ada, susah. Kan? So your skin is protein, yeah, it's a specific type of protein. We call it as keratin, yeah. So kalau macam kulit you kering, you nampak kan kulit you kering macam tu ada putih-putih, that's keratin. Yang kalau dia dia kena bertukar ya, especially like you, yes, you are still young, so your skin bertukar, so your skin will be released in form of keratin ya, the outer layer of your skin. So kalau that's why if you're wondering dalam your room, you know, tak ada orang masuk, you just orang, tapi tiba ada habuk. Habuk dari mana? You tutup tingkap kan, tutup pintu, tiba ada habuk. Habuk comes from your body lah, from your skin ya, because your skin will release the dead cell, the dead skin ya. Especially when you stay in air conditioned room, yeah, and your room is very dry, or if you live in four season countries, during winter your skin will become very dry. So that's the time that your skin will release a lot of dead cells, dead skin cells that consist of keratin. Yeah, and in most cases, yeah, just like happened to me 15 years ago when I work in the US. Yeah, so I work in the lab during winter. Yeah, and winter in Kansas, the state that I went for work, yeah, for my postdoc. Sometimes during winter the temperature can drop to minus 30 degrees Celsius. 30 ya, bukan 13, 30 ya, minus 30 degrees Celsius. We still have to work. No excuse. Tak ada excuse, ya. We still have to work. We still have to walk ya, to the lab ya. Very thick snow, you know, 1 meter, 2 meters snow. We just have to walk, you know, bear the bear the wind, bear the cold, you know. The skin, our skin very dry, and it happened to me that it contaminated my sample, you know. So I work with cell culture, yeah, with cancer cells. Yeah. So it contaminated the cell, yeah, because winter, you know, kulit kering, garu-garu kan, garu sana, garu sini, rambut pun tak potong, you know, garu sana, garu sini. So the keratin, the protein from my skin, fall into my sample. When I send my sample for analysis, they discover a lot of keratin. I thought, well, maybe the cancer cells contain keratin, and the whole lab laughed at me. Ha 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 ha. No, it's not true. It comes from your skin and your hair, you know. Because, well, during winter, you know. So yes, it will contaminate. So that's why, you know, in some labs when they work in the lab, they wear like astronaut, like the special dress like astronaut, you know, to avoid the keratin from contaminating the sample. Yeah. Okay, so is my voice still clear?
Yes, doctor.
Okay, all right. Okay, thanks. Yeah. Just so first, I want to make sure that the connection is okay. Second, to make sure that you are still awake. Yeah. All right. So the second one is globular protein. Globular protein is the hydrophilic. Yeah. So they are soluble in water. Yeah. It consists of enzymes, you know, regulators. They are dissolved. Yeah? They are soluble in water. Yeah. So the shape is like is round. Yeah. It's like globe. Yeah. So that's why it's called globular protein. Yeah. So it's spherical. Yeah. The shape is spherical. So mostly it's water soluble. Yeah. So these are some examples of fibrous protein yang dia water insoluble. Yeah. Keratin, collagen, silk, they are keratin as well. And then, so these are some properties of fibrous protein. Yeah, all fibrous protein are water insoluble. Yeah, alpha keratin in your hair, in wool, in nail, you know, in horns, in tusk. You know, have you read the research paper that now we have more and more elephant without tusk? Yeah, without apa in Malay gading, gading, without tusk, tuskless elephant. Somehow it's because of evolution. Evolution forced the elephant to produce more and more tuskless elephant, because they know, somehow evolution knows, yeah, that when you have tusks, when the elephant have tusks, then they will be killed. They will be killed by hunter, yeah, to get their tusks. So it's amazing that evolution now force elephant to be tuskless elephant. They don't grow tusks anymore. Somehow their hormone change, because what? Because gajah jantan je yang dia ada tusk kan. So somehow their hormone change. Dia jantan tapi dia punya tusk tak grow. Yeah. The elephant is male elephant, but somehow their tusk, they don't grow their tusk. this is amazing to see that human actually can force the direction of the evolution. It's amazing, but it's sad at the same time that because of us, yeah, because of our act of killing the elephant, they are forced to not to grow their tusks, yeah, their guarding, yeah. So it's quite sad actually, because the male elephant with lower hormone that control the growth of tusks, they don't have long lifespan, yeah, their lifespan becomes shorter, right? And for your information, human is the most destroying animal on earth. We are the most destroying. We are we are the one who is responsible for the extinction of a lot of species. Yeah, especially after the Industrial Revolution. Yeah, so we are responsible for the climate change. Yeah, nampak sekarang masih panas ya kan. We are responsible for the extinction of plenty of species. I have the data. Then when I saw the data, I feel very sad, you know, that yes, hundreds and even thousands of species extinct because of human, yeah, and we don't do any corrective action, yeah. This is the only Earth that we have. Yes, we are thinking of moving to Mars, to Moon, but there is still far away, you know. That possibility is still far away. The only thing we can do is to keep this Earth. So because this is the only Earth that we have. You know, there is no other Earth that we know with the perfect condition that we can live. You know, so yeah, you know, now we have more and more elephant without tusks. Yeah, without dia tak ada gading, even though they are jantan. Yeah, so that's how evolution works, and it's quite sad to see that. All right. Yeah, so this is the structure of your hair. Yeah, if you're wondering the structure of your hair, this is the structure of your hair. So it's a keratin, yeah, but somehow it has an alpha helix structure, yeah, even though it's fibrous. And if you want to see more detail about your hair, this is what you have in your hair, lah. Yeah. All right. So this is your hair. So kalau you cut the cross section of your hair, you will see that this is the keratin. Siapa yang suka nak tukar hairstyle tu kan? This is what happened lah. Actually, it's a chemical reaction. Yeah, chemical reaction. Yeah. Contohnya kalau rambut you straight kan, rambut you straight, you nak buat wavy sikit, then what you have to know is the style of your hair is maintained by the disulfide bond, yeah, between cysteines, yeah, between cysteine amino acid. Yeah. And this covalent bond can be broken, yeah, can be reduced by reducing agent, yeah. And reducing agent yang selalu kita guna ni selalunya berbau. That's why kalau you pergi mall, you jalan lalu semua hair salon semua, you ada bau kan, you ada rasa macam bau busuk kan, because of the reducing agent that they use, because of chemical they use to break the disulfide bond, yeah, to break the disulfide bond. So if you're wondering why it's always smelly, yeah, if you work in the lab like me in the organic synthesis lab, you are familiar with the smell. Selalu ada. Dan kita pun yang semua yang buat kerja dalam lab tu berbau lah. Masa kita keluar dari lab, kita pergi balik rumah memang bau lah. So the smell that you sense, you detect whenever you walk through a hair salon, you know, because of the chemical they use to reduce the disulfide bond, yeah, in your hair. And then lepas semua disulfide bond is broken, yeah, you reduce into SH. And then you curl them, yeah. You curl them into the shape that you want, and then you oxidize back using oxidizing agent, yeah. And then they will create a new disulfide bond, yeah. And because this is a covalent bond, your hairstyle can be maintained in a very long time, yeah. Because this is disulfide bond, yeah. Disulfide bond is difficult to break, okay. So your hairstyle your new hairstyle will stay lah. Nanti nak tukar balik, pergilah hair salon, dia akan bubuh chemical, the same chemical to reduce into SH, and then boleh tukar style rambut yang baru lah. So there's a chemical reaction. So kalau you pergi hair salon, tukar rambut, you explain lah to your hairdresser, oh, this is the chemical reaction that happened. So explain to them. So right. So this is collagen. Kita ada collagen in our connective tissue, in tendons, cartilage, bones, cornea of the eye. So each collagen chain is a long glycine and proline-rich left-handed helix. So and it's quite strong. It's quite strong. So that's why collagen can be found in connective tissue. In tendons, cartilage, bone that support our body, because they have strength. They have certain strength that can maintain the integrity of our body. This is the structure of collagen. If you're wondering, macam ni. This is the structure of collagen. So almost similar to your hair. And this is the structure of collagen under the microscope. And then silk, silk, mainly beta structure, antiparallel beta structure, and silk is very strong. Silk is very strong. The same amount of silk has a stronger tensile strength compared to metal. It's very strong, basically. So it's amazing how a very tiny creature, spider, can produce something that even stronger than steel. Nampak kan Spider-Man kan, actually it's true. The silk that he released, can be as strong or even stronger than metal. So it's true. Maybe you can use genetic engineering, gene engineering to create a Spider-Man in the future. Because I told you last week, one of you asked me, is that possible to change our gene, using some genetic engineering? It's possible. I told you, right? I told you that in China, we have 2 babies, product of gene engineering, So in the future, you want to create Spider-Man, real Spider-Man, that can produce all this silk from his hand, tembak sana, tembak sini, So it's possible, why not? So in science, imagination has no limit. Imagination has no limit. So in the past, people imagine, how can we modify our gene? Now we can do it. Even now, it's a very normal procedure in the lab. It's very normal procedure in the lab. So that's why we come up with different, genetic modified organism. Because that's normal way, we do it in the lab, all the time. So and then people go to Mars, something that in the past it was just our imagination, that because of the power of science, now it becomes reality, including, to come up with a group of Avengers. Why not? Now we can change the gene. Maybe one day, a human can produce silk just like Spider-Man, boleh lekat sana lekat sini kan. Why not? All right, so this is the structure of silk. So it has an anti-parallel structure. Globular protein, so mostly soluble, and these are the proteins that we have in our body, inside our body, in our blood, in our organs. Kalau yang dekat luar ni mostly insoluble, rambut, kulit kan. Kalau yang dekat dalam, dalam darah, dalam serum, your antibody, all are soluble protein, and they are globular protein. Myoglobin is one of them. Myoglobin is the protein responsible for delivering the oxygen from your lung to muscle. It's small protein. Dia punya size is only around 16,700. But they work as tetramer. So that's why even though one unit is small, but they join together, tetramer, so it becomes big. So as I mentioned, the job of myoglobin is to bring oxygen from your lung to muscle, yeah, and then bring carbon dioxide from muscle to lung. So dia menjadi macam grab lah, bawa sini bawa sana kan. So this is the structure of myoglobin lah. So nanti oksigen dia akan bind dekat tengah ni, ya, dia akan bind dekat tengah. So it will be transported from your lung to cell. So it cannot it doesn't only bind oxygen, it can bind something else as well. It can bind cyanide. Cyanide. It can bind cyanide with even stronger binding. So that's why kalau you nampak dalam cerita movie kan, Nazi dulu kan, masa apa, the Allied forces tangkap diorang terus ambil cyanide pills to kill themselves, because cyanide can bind even stronger to myoglobin, so they become oxygen deprived, so they die quickly, because the binding between cyanide and myoglobin is very strong, stronger than oxygen, so oxygen cannot bind anymore to myoglobin. So cyanide. And then another thing that can bind to myoglobin is not only oxygen, cyanide, carbon monoxide. Carbon monoxide, Carbon monoxide or CO can bind to myoglobin stronger than oxygen. Tu ada kes-kes macam orang tiba-tiba mati dalam kereta kan, sebab engine dia bocor, because the engine is not well maintained, tak pernah bawa servis, malas bawa servis kan. So the oxidation process, instead of producing carbon dioxide, the engine, car engine produce carbon monoxide. Lepas tu bocor, masuk dalam aircon, you tidur, berdating kan, berdating dengan awet dalam kereta, tak sedar, terhidur carbon monoxide, terus tertidur. You don't feel pain, terus tertidur. Pengsan. And then die. Alright? So kalau berdating tu open space lah, jangan dalam kereta. Okay. Alright, atau dalam kereta tapi tak payah buka aircon, panah. Right, and then okay, in other organisms, in human, dalam myoglobin ni kita ada ferrum, iron, Fe, and Fe is the one who is responsible to make your blood red. Itu yang darah you warna merah. Sebab dalam your myoglobin dia ada ferrum. Ferrum color dia merah. In certain organisms, instead of ferrum, instead of iron, in their globin, they have other metals. Copper. Copper. Ini pun boleh bind oxygen juga, copper. Color dia biru. That's why certain organisms, warna darah dia biru. Because of what? Because instead of ferrum, in their globin, they have copper. To bind oxygen. And then ada yang nickel, Ni. Color Ni is hijau. Certain organisms, darah dia color hijau. Kita somehow by design ferrum, so darah kita warna merah lah. Start lah you fikir kan, how do we do genetic engineering to change our blood color to blue? Possible. You just have to do some genetic engineering so that our myoglobin, instead of having Fe, we have copper, so your blood becomes blue, blue blood. Okay, lain sikit kan. But merah okay lah, dah biasa kan dengan color merah kan. But it's true, so the color of our blood comes from ferrum, from the iron that we have in our myoglobin. So this is the structure of myoglobin in different way of drawing. So if you have a big secondary structure, it becomes motif. It becomes motif. So motif is called super secondary structure. For example, if you have a protein, all alpha helix or all beta sheet or both. So for example, this one, semua beta. So kita panggil this motif as beta barrel. Certain proteins, they have this motif. We call it as beta barrel. Or this, beta alpha beta loop. This is motif as well. Or complex motif. Beta alpha beta loop or alpha beta barrel. So because protein, most protein, they have more than one secondary structure. Dia ada beta, dia ada alpha. So the way alpha and beta arrange, or the way beta, beta, alpha, alpha arrange, will give you a motif. Yeah. So different proteins, sometimes they have the same motif. Yeah. Sometimes. Okay. And then protein quaternary structure. Yeah. So for example, myoglobin, as I told you before, myoglobin works by combining 4 subunits into one. Yeah. So ini kita panggil as protein quaternary structure lah. So only in multi-subunit proteins, not in all proteins, only in multi-subunit protein. Okay, so I skip this. All right, protein stability and folding. Yeah, so we talk about protein structure, and then I explain a bit about Moderna vaccine, Pfizer vaccine, how they stabilize the protein. So this is very important. Yeah, this is very important. How to stabilize the protein structure. Yeah, because the function depends on the structure. Kalau structure dia tak stable, function dia hilang. Sama macam mRNA vaccine. Kalau structure dia bertukar daripada pre-fusion kepada post-fusion, dia punya function hilang. Not only that, I explained yesterday, we saw this in RSV vaccine, when they use post-fusion conformation, instead of blocking the virus, the vaccine actually helps the virus to infect human. So the function of protein really depends on its structure. Sangat bergantung kepada dia punya structure. So now the question is, how to stabilize the protein. And you have to know what factors that can destroy the protein structure. Most proteins lose their structure because of denaturation. So this is the term that you need to remember. Denaturation. Denaturation. So basically, the protein lose its structure. Right. So it could be caused by several reasons. Yang pertama, panas. Heat. Heat could destroy the protein because it will destroy the non-covalent bond. I explained yesterday, the protein structure is maintained by weak interaction, hydrogen bond, London dispersion, hydrophobic interaction, where all this weak interaction can be destroyed by heat. You increase the temperature from 37, normal body temperature, to 40, all this interaction gone already. So temperature can destroy the protein. Not in terms of destroying the covalent bond, covalent bond is still maintained, but they destroy the protein in terms of the weak interaction dia hilang. That's why this is one way to kill the virus lah. Kalau sekarang COVID kan, you nak tahu macam mana nak kill the virus, guna heat lah, guna heat. That's why it's good idea not to be in a closed space. It's a good idea to be in open space, because heat will kill the virus. It will destroy the protein on the virus. And then extreme pH. Extreme pH will destroy the protein as well by disturbing the electrostatic interaction between charged amino acid. So beberapa amino acid, they have charge. Arginine, lysine, aspartic acid. If you change the pH, their charge will change, and as a result, the protein structure will change. So you can destroy the protein because of pH. You tukar pH 1 unit, it will destroy the protein structure, and the function will be gone lah. Organic solvent, detergents, it will destroy the protein as well, because they disrupt the hydrophobic interaction. That's why masa COVID ni, you kena rajin basuh tangan. You kena rajin basuh tangan, because when you use detergent or use organic solvent like alcohol, it will destroy the protein on the virus. It will destroy the hydrophobic interaction that stabilize the viral proteins. Simple je basuh tangan. Tak perlu pun basuh tangan dengan sabun yang ada disinfectant, tak perlu. Any soap, ya, any soap, ya, tak perlu nak ada macam-macam chemical. Any soap, because you know that the detergent will destroy the hydrophobic interaction that stabilize the viral proteins. Yeah. Organic solvent that we use is ethanol, 70% above. Eh? And then cotropic agents, urea and guanidine hydrochloride, will also disrupt the water molecules that solvate the protein. Because you know, I explained yesterday that this is the protein, and the protein is surrounded by water, kan? Surrounded by water. So this arrangement stabilizes the protein. But when you add urea and guanidine hydrochloride in high concentration, 6 molar, it will strip off the water molecule. Yeah. Dia akan tukar water molecule ni, dia akan buang water molecule ni, so protein structure will be destroyed. Yeah. Dia akan terbuka. Yeah. Alright, so far my voice still clear, eh?
Yes, clear.
Alright. Alright, so yeah, luckily the connection is good today. So this is what happened with two proteins when you change their temperature. Yeah, this is the percent unfolded. Yeah, so the percent unfolded, you see that this is the first protein, ribonuclease. When you increase the temperature, yeah, above 60 degrees Celsius, almost all ribonuclease are destroyed. Yeah, almost 100%. Apomyoglobin, yeah, this is the protein in your hemoglobin. Yeah, so at around 65 degrees Celsius, 50% destroyed. Yeah. You increase to 100, around 85%, and around 90% of protein will be destroyed. So that's the effect of temperature. Yeah. So this is the effect of guanidine hydrochloride. Yeah? We call it as a caltropic agent. Yeah? So when you increase the concentration of guanidine hydrochloride, yeah, above 5 molar, all the proteins will be unfolded. Lah. Dia akan, the structure will be damaged. Yeah? In most cases, we just put 6 molar, yeah? in just to make sure that all proteins are destroyed. If we want to see the effect of guanidine hydrochloride to the structure of protein. Some proteins, they can get their structure back. Yeah, if the destroying agent is removed. Yeah, macam tadi kita letak guanidine hydrochloride, right? 6 molar. Tapi kalau kita remove slowly, some proteins can go back to their initial structure. Yeah, contohnya ribonuclease A. Yeah, dia boleh dia boleh balik ke structure yang semula lah. So this is the initial structure, yeah. And then kita letak urea or mercaptoethanol. This is the unfolded structure. We call it as a denatured structure. Tapi kalau kita buang, ya, kita remove urea and or mercaptoethanol, ya, dia boleh balik ke structure yang semula lah. Ya. And this has been proven by Christian and Vincent. Ingat lagi kan? Christian and Vincent is the person who says that the structure of protein depends on the sequence. So that's why he came up with that concept. Yeah, we call it as Anne-Vincent dogma. Ingat lagi kan my first lecture, ya, Anne-Vincent dogma, because of this experiment. Yeah, so he took this protein and then he destroyed the sequence. He destroyed the structure, the three-dimensional structure of protein. Lepas tu dia slowly remove the destroying agent. Dia buang-buang with dialysis lah. Ya, dia buang-buang. And then tiba-tiba dia realize, the structure, the protein goes back to the initial structure. Maknanya apa? Maknanya the structure of the protein depends on the sequence. If the sequence is still there, when you remove the destroying agent, when you remove the denaturing agent, the protein will go back to initial structure. Yeah. So protein misfolding is the basis of numerous human disease. Yeah? I explained to you about Parkinson's disease. Parkinson's disease, yeah, is caused by the you know, the tau protein, yeah, the protein that keeps the integrity of your nerve cell. Masa you dah tua, the protein becomes misfolded, yeah, because of hyperphosphorylation, yeah? and it causes Alzheimer's disease, yeah, in human, yeah. Cystic fibrosis as well, it's caused by mutation in the gene for the protein cystic fibrosis transmembrane, yeah, and the mutation in this protein causes misfolding, and it's not treatable, yeah? It cannot be treated, yeah? Once you get cystic fibrosis, then you have to live with that disease for the rest of your life. Mad cow disease, ya, caused by misfolded protein. Ini berlaku dekat lembu, ya. So because of that protein, the misfolded protein in their brain, the cow become mad, ya. So the cow become mad. So if you open their brain, you will see some protein are misfolded there. So that's why the cow become mentally disturbed. Yeah. Yeah. So I mentioned a bit about this, yeah, when in my introduction, in my introductory lecture, that the folding process has to be assisted. Some proteins, but not all, they can fold spontaneously. Keluar dari ribosome, they can fold spontaneously. Yeah, boleh. Yeah. But most proteins, in order for them to fold, you have to help them using assisted folding. Yeah. So the protein that helps other protein to fold is called chaperone. Yeah, they call chaperone. And there are two classes of chaperone. Yeah, heat shock protein, we call it heat shock protein, or chaperonin. Yeah. So macam ni lah. Yeah. Cara apa? One example. Yeah, I put here how heat shock protein. Yeah. HSP70 helps the protein to fold. This is another another picture how chaperonin helps the protein to fold. So protein masuk, yang baru keluar dari ribosom, dia masuk dalam ni, lepas tu dia tutup, pop, kacau. Lepas tu dia keluar dari sini, dia dah folded. So dia macam kilang, raw materials masuk, dia tutup dengan grow S, dia masuk dalam grow L, tutup dengan grow S, kacau, keluar, protein dah siap, dia dah folded. So this is the atomic structure of grow L and grow S. This is grow L, where the protein is folded. This is grow S, dia punya cover lah. Lepas protein masuk dalam ni, dia tutup. Lepas tu the folding process happen. Lepas tu cover ni buka balik, protein keluar, dia udah folded, dah kemas, dia dah dilipat. That's all for today. Yeah, I think we finished this topic, about protein structure. Next week I will continue with protein analysis. How we analyze the protein structure and whether the protein is there in our sample. Yeah. Any question? Any question?
For now, no questions, Doctor.
Okay, no question. Semua dah isi attendance. Tak boleh lupa the first 15 minutes. First 15 minutes you have to fill up the attendance.